Hello, this lecture is intended for my students taking A&P 2 at Madison Area Technical College this spring of 2020. The focus will be on the second and third processes that occur during urine formation. If you recall, the first process is glomerular filtration, shown by the red arrow here in this diagram. If you need further information about nephron function, I have posted some other YouTube videos. And in addition, you can use your reading assignment. And for details about what you should be comfortable with, check study question 15. Before we get into too much depth, I want to make sure the definitions of reabsorption and secretion are clear to you. Reabsorption means that substances move from the nephron back into the body, which means into the interstitial space, that means between the cells in the kidney, and then into the bloodstream. So substances that have been filtered out of the blood can re-enter the blood through the process of reabsorption. It's a way for the body to recapture what's been lost during filtration. And since it's the cells of the nephron tubule that actually do this first transport process, we call it tubular reabsorption. Secretion, or tubular secretion, means that substances move from the interstitial space into the nephron tubule. Again, it's named in reference to the nephron tubule because eventually substances will have to cross the tubule wall. There are various routes that substances use to get in or out of the nephron. Here I show two routes of tubular reabsorption. In particular, secretion basically would just happen in the opposite direction. So the tubule lumen is on your left. That's where the urine is being formed, and you'll see that there's water and solutes there. Then we have the wall of the nephron, made up of simple cuboidal epithelium, and so I've got it labeled tubule cell. Then in between the nephron and the capillaries surrounding the nephron, there's space, fluid. We call that interstitial fluid. And then finally on the right, you see the capillary. So substances can take two routes, one of two routes. Either they go through the cell, which is shown by the transcellular route right here. That might be right across the cytoplasm of the epithelial cell. Or sometimes they take a little detour and at the very end go in between cells. But either way, they've had to go through the cytoplasm and across this apical membrane in order to get out of the nephron tubule. The other route is called the paracellular route. Substances move across the wall of the nephron in between two cells. So that happens with water and solutes as well, and we'll talk about why and how in just a little bit. First, a little bit more overview. 99% of filtrate is reabsorbed. What gets reabsorbed and how much gets reabsorbed depends on the body's need for water, certain electrolytes, and acid-base balance or pH balance. Almost all of this filtrate reabsorption occurs in the proximal convoluted tubule, 65%. The main driving force is sodium being actively pumped from the tubule lumen back into the body or into the blood, and water follows. 25% of reabsorption occurs in the loop of Henle, and then the remaining 8 to 9% is reabsorbed in the distal convoluted tubule and the collecting duct. Secretion really doesn't have much impact on urine volume, but it is going to be important, particularly in getting rid of some wastes that were not filtered, maybe wastes that weren't even in the bloodstream, they're in the interstitial space, so they need to be secreted into the nephron tubule. We get rid of some wastes that are byproducts of metabolizing drugs, pharmaceuticals, this way, as well as some salts and hydrogen ions. Secretion primarily happens in the proximal convoluted tubule, but also in the distal convoluted tubule and the collecting duct. So let's start with the proximal convoluted tubule and look at what's reabsorbed there. Sodium and water are the main things that we are concerned about, but also some nutrients that might be lost in the urine if we weren't able to recapture them. So first I'll deal with sodium. Sodium is reabsorbed by two processes: primary active transport and secondary active transport. I'm going to show you pictures of those in a moment. Water usually follows sodium by osmosis. So where sodium goes, water follows, and water can move through pores in the cells called aquaporins. Because sodium is actively reabsorbed and water must follow, we call this water reabsorption obligatory. Water is obliged to follow sodium. Then if the filtrate contained any glucose that was in the blood, or maybe amino acids, both of which are organic nutrients, the body wants to recapture those, so they are reabsorbed, and the process is secondary active transport. Interestingly, they generally are transported with sodium, so I'll show you pictures of those as well. So here are the mechanisms, the transport mechanisms for the reabsorption of sodium, glucose, amino acids, and water. First, labeled number 1 in green, shows active transport by the sodium-potassium pump. This is on the basal surface of the cell, the nephron wall, I guess, close to the interstitial space, and 3 sodiums are pumped out of the nephron tubule cell, and 2 potassiums go into the nephron tubule cell. So this helps move sodium against a gradient. So that means we use ATP to create a gradient of sodium. The gradient will be such that sodium will be low inside the tubule because it's being pumped out. So then let's look at number 2. This is on the other side of the wall, close to the lumen of the tubule. And here is a transporter. It's called a co-transporter. And what it does is it allows sodium to diffuse down a concentration gradient. So the fluid in the lumen has high sodium. Sodium will move down its concentration gradient, which will help facilitate the sodium-potassium pump as well. But it's the sodium-potassium pump that created this gradient. That's why we call this secondary active transport. The movement of sodium did not require ATP here at number 2, but it needed ATP to create a gradient via the sodium-potassium pump. When sodium moves down its gradient, it's kind of like providing energy for other substances to piggyback with it. And this is how organic nutrients are recaptured from the tubule fluid. So here we have glucose and amino acids that are absorbed with sodium. They enter the tubule lumen, and by another transport process they're going to move out of the tubule cell, out of the tubule cell, and then eventually into the blood, but we're not going to cover that. Now, as I mentioned, water always follows sodium. So here's water, number 3, shown in blue. Water will go through aquaporins by the process of osmosis following solutes. Lipid substances can cross cell membranes. They like phospholipids, so they'll just diffuse down a concentration gradient. That would mean if they are high in the fluid, like a steroid hormone or something small, then it'll cross over into the tubule cell and eventually reach the blood. The last transport mechanism for reabsorption is sometimes called solvent drag, and this involves substances moving with water through by a transcellular route, meaning in between cells. And it's kind of just due to hydrostatic fluid pressure. So there's a lot of fluid in the tubule lumen, and some pressure is building up, not a lot, but some, and that creates pressure or a force to move water in between cells, and with it water brings ions and urea and other substances. Now the blood pressure in this capillary is kind of low, 8, I'm trying to write with crayon basically, 8 millimeters of mercury. That's low pressure. And so if there's higher pressure inside the tubule lumen, that'll draw water to enter the capillary. This is a picture of how sodium and glucose are co-transported during reabsorption. The problem with this picture, I kind of like it because it's really clear, but the problem with the picture is now we've reversed. So this is where the tubule lumen is, over here, the lumen. Okay, that's really sloppy. And this is where the blood is, over here. So sodium is going to move back into the blood, bringing with it glucose. And this is the transporter that's used. It's called a sodium glucose transporter, has an abbreviation. It's also sometimes called a symport because two things are moving simultaneously. Now, if glucose has been filtrated at the glomerulus, it's nice that it can be reabsorbed, but it is limited how much can be reabsorbed. That's called a transport maximum. So we need to talk about that next because it's kind of related to diabetes, and you'll be interested in that part. So a transport mechanism, by definition, is the maximum rate at which a substance can be reabsorbed by a particular protein transporter. The reason there's a maximum and reabsorption isn't unlimited is because these transporters are saturated. For glucose, the maximum is 320 milligrams of glucose every minute. So if there's more glucose filtrated, or at least if it's filtrated faster than that, then not all of the glucose can be reabsorbed, and then you'll have glucose in the urine. Water follows glucose into the nephron or wherever its high concentration is. And when water follows glucose into the nephron, you'll have more urine volume. So this is a classic sign of diabetes. You'll have glucose in the urine because the transporters can't reabsorb it quick enough. And you'll have polyuria because water has followed the glucose into the urine. Polyuria meaning a high urine output. So the picture on the left shows normal blood glucose levels, and the glomerulus is shown different color. Try this color. glucose is being filtered from the glomerulus into the nephron. Normally, in the proximal convoluted tubule, glucose can be reabsorbed. So that's reabsorption. But what if blood glucose levels are high? So hyperglycemia. Now more glucose is filtered or pushed out of the bloodstream, but the transporters cannot keep up. Therefore, you're going to have glucose in the urine. And because water follows, you'll have polyuria. I hope that made sense to you. Okay, there are some substances that are secreted into the proximal convoluted tubule. The one that I really care about the most is hydrogen ion, because we will address that again later. Hydrogen ions, of course, determine pH, or the concentration of hydrogen ions do. And when the concentration is high, the blood pH is acidic. We can get rid of these excess hydrogen ions in the proximal convoluted tubule as we reabsorb sodium. And I'll show you that process. There are other things that are secreted into the proximal convoluted tubule. Certain drugs that don't get filtrated because they're bound to large plasma proteins. We can get rid of those, like methotrexate, penicillin, morphine, histamine, dopamine. So the kidney will actually secrete those materials into the nephrons or urine forming areas. And as I mentioned before, some nitrogenous wastes that weren't even in the blood perhaps, they might be in the interstitial space, they can be pumped into the nephron as well. But like I said, I'd like to focus on hydrogen ions, so let's look at what is used for hydrogen ions. It's again a transport protein, a pump if you will, or a transporter, but this time it's an antiport with sodium. So notice that as sodium is reabsorbed, so sodium is going to get reabsorbed. This is the urine that's forming over here. Oh, I cannot do this. Let's try that again. So here's the urine. And in the urine is sodium. So sodium is going to be reabsorbed. We already know that sodium can be reabsorbed with glucose. And of course we've got the sodium-potassium pump over here. But in this instance, hydrogen ions are moved in the opposite direction when sodium is reabsorbed. So this is secretion. That's what that is. It's secretion. Hydrogen ions can be secreted out of the blood and into the nephron tubule. So a summary of what happens in the proximal convoluted tubule is that we get a lot of filtrate reabsorption. In particular, water, and some sodium, and some glucose, amino acids if they're there. And I didn't mention this, but this is another place where we can deal with acid-base balance, and that's by reabsorbing bicarbonate, which is a base. Substances that are secreted at the proximal convoluted tubule that we really care about are acids, so hydrogen ion concentrations. Next we'll look at the loop of Henle. Okay, at the loop of Henle both reabsorption and secretion take place. Starting with reabsorption, 25% occurs here. And what is reabsorbed are both ions and water. The secretion that happens is usually a nitrogenous waste product in the interstitial fluid called urea. And urea is the precursor to urine. So urea has amine groups. It's not working. Okay, that didn't work. Sorry. All right, so the descending limb of the loop of Henle is permeable to water, but not to any ions. And it's only permeable to water in this thick portion, but you don't have to worry about that. I don't need you to know that detail. The ascending limb is permeable to sodium, potassium, and chloride ions, but it is not permeable to water. So you might ask, why does this occur? Well, together they form something called a countercurrent multiplier. That's just a fancy term for saying that because fluid is moving in opposite directions in the two limbs, we can maximize the reabsorption of solutes, and that means that the renal medulla, where the juxtamedullary nephron loop of Henle is, becomes very concentrated. And it's really important for blood volume because we can reabsorb more water from the collecting duct then. Let's look at how this nephron loop works before we talk about the advantages of it. Okay, so the transport processes in general would be osmosis for water and active transport for the ions. And when urea is secreted, it's facilitated diffusion, although I will be ignoring that to some extent. So this loop, because of its selective permeability and the fact that the fluid is moving in opposite directions, creates an osmotic gradient in the renal medulla. So notice in this diagram, the cortex of the renal medulla is 300, has an osmolarity of 300. This is the cortex. My pen isn't working very good. That's the cortex. And at the central or deepest part of the medulla, it can get up to 1200 milliosmoles. That varies depending on the body's current water retention or elimination processes. And the variation allows the kidney to produce urine that is very concentrated relative to our normal body fluids, or very dilute compared to our normal body fluids. So hypertonic or hypotonic. And it does, this loop creates this gradient, and then the collecting duct has fluid that moves through this gradient. And as the fluid is exposed to more highly concentrated environments, water can be reabsorbed. This is how we maximize our water reabsorption. Okay, let's look at how this osmotic gradient is created. We need to start primarily, I think, over at number 1 in the ascending limb over here. Salts are pumped out of the ascending limb. That's active transport. Sodium, potassium, and chloride. So that's making the interstitial fluid highly concentrated. Secondly, we have new filtrate that comes in, and it's rather dilute to begin with, much like the blood, although it doesn't have plasma proteins in it. And so it's even more dilute. And as this dilute fluid enters the descending limb, it's exposed to this concentration of ions, and that makes water be reabsorbed. So water's reabsorbed. The filtrate then that's entering this kink in the loop, I'll call it, is now concentrated, highly concentrated, saltier. It's lost water. Well, that works well because the next part of the nephron is going to be able to take that salt and pump it out into the interstitial space. Therefore, the fluid that leaves the ascending limb is dilute. So that's dilute, relatively dilute. It's lost salt, it's lost water, then it lost salt. And now the collecting duct is carrying this dilute fluid, and it plunges into the medulla of the nephron, where it's exposed to a high concentration of ions, and water is reabsorbed. So the fact that the loop creates the gradient and then the collecting duct takes advantage of the gradient is what gives us the ability to create what is called a hypertonic urine and promote water reabsorption. There's one more factor that promotes water reabsorption, and that is that between the loop of Henle and the vasa recta capillaries, there is a countercurrent exchange mechanism. What that means is that when the nephron, or where the nephron fluid is traveling in a particular direction, the blood is traveling in the opposite direction. That's the countercurrent business. So in the descending limb, we've got fluid moving, I'm going to say downward, and the vasa recta is moving upward, that part. When blood enters the vasa recta, it enters in capillaries close to the ascending limb of the vasa recta, which is carrying tubular fluid in the opposite direction. So on both sides you have this countercurrent exchange. What that means is that the blood that has been filtered and now is entering the vasa recta is relatively dilute, but it is entering this highly concentrated region, and that makes it lose water. So it loses water. But when it rounds the bend here, like this, it's still more concentrated than the interstitial space, and so more water is reabsorbed here. You don't really have to understand the complete details and the numbers, although I did post a video so that you can see how it works. But what's most important to know is that the anatomical arrangement and this countercurrent flow of two different fluids really promotes water reabsorption. Okay, let's look at a summary of the transport processes we've talked about so far. We know in the proximal convoluted tubule, water, sodium, and glucose is reabsorbed, as well as amino acids, and hydrogen ions are secreted, so we can get rid of those. We also know that in the loop of Henle, water is reabsorbed as well as ions, which I'll call salts. That's the most important part. Now we need to go to the distal convoluted tubule and see what that can do for us. So the distal convoluted tubule is also involved with reabsorption and secretion, but to a lesser extent. This region of the nephron can be controlled with hormones. Its permeability is controlled by hormones. What's reabsorbed is sodium, calcium, and water, and what's secreted is potassium. These things are hormonally regulated. There's one hormone, aldosterone. You might remember aldosterone. That's involved with the reabsorption of sodium and at the same time secretion of potassium. They're linked. Water always follows sodium. So aldosterone allows us to reabsorb water because water follows sodium. And for calcium to be reabsorbed and kept by the body, there's a hormone that regulates how permeable the nephron is to calcium, and that's parathyroid hormone. Let's look at a picture at how sodium and potassium are linked in terms of sodium reabsorption and potassium secretion. So this is the first picture we started out with transport processes, and this shows the location where sodium is pumped by the sodium-potassium pump back into the blood, we'll say, and potassium goes in the opposite direction. On the other side, other surface of the tubule cell, we have sodium and potassium antiport. So the sodium, it's kind of like the one with hydrogen ions. Actually, it's exactly like that. So sodium is going to be reabsorbed from the filtrate, and potassium at the same time is going to be secreted into the tubule lumen. The gradient for sodium to be reabsorbed in this way is created by this sodium-potassium pump. In the collecting duct, we have two hormones that are particularly important. One is aldosterone and the other is antidiuretic hormone. Both of them contribute to water reabsorption. So water is reabsorbed in the collecting duct by the process of osmosis, because the collecting duct is moving into the deep renal medulla. And sodium reabsorption is happening, which just promotes more water reabsorption, and that is by the same mechanism that happened in the distal convoluted tubule. There's another factor, though, that plays a role, and that is the hormone ADH. There's a delay here. ADH. ADH is antidiuretic hormone, and antidiuretic hormone just promotes the number of aquaporins that are present in the convoluted tubule. So there'll be another aquaporin, another one put in, and so it just promotes water reabsorption. I put this slide as a Word document on Blackboard because I think it's a good way to study where substances are reabsorbed and where they're secreted, is to make yourself kind of write it over and over again. So you can print out that document and play with it, or draw your own nephron and mimic this particular picture, if you will. So the next slide just shows you a summary of what I mentioned about hormones, but then I added a couple here too. The two biggies are aldosterone and antidiuretic. So aldosterone acts on the distal convoluted tubule and the collecting duct. It increases the production of sodium-potassium pumps so that sodium can be reabsorbed and then water follows. In the process, potassium is secreted because it's a sodium-potassium pump. Then I'm going to jump down to antidiuretic hormone. It acts on the collecting duct because it stimulates the production of aquaporins, and that increases water reabsorption. There is another hormone that I have not mentioned for a long time, maybe in the circulatory system, and that's atrial natriuretic peptide, ANP. ANP inhibits sodium reabsorption from the collecting ducts. It actually antagonizes the effects of aldosterone. And angiotensin II, you may remember, stimulates aldosterone production. So if we had high filtration or low blood pressure, I should say, in the glomerulus, remember renin is produced, and renin can convert angiotensin I, is involved in the production of angiotensin II, I should say, and then aldosterone is produced. So this is a summary slide of everything to do with reabsorption by the nephron and secretion. Notice that everything that's in blue is reabsorption and everything that's green is secretion. 65% of reabsorption happens in the proximal convoluted tubule, including sodium, water, and those organic nutrients. Hydrogen ions can also be secreted into the nephron tubule, as will potassium, not listed there. The nephron loop is involved with reabsorption of water and ions, sodium, potassium, and chloride. It creates a gradient in the renal medulla so that the deeper you get into the medulla, the more concentrated is the interstitial fluid. And the collecting duct dives down into that environment, and water is reabsorbed. But I've skipped the distal convoluted tubule, so let's talk about the distal convoluted tubule. Is where sodium and calcium can be reabsorbed, but that's based on hormones, so it's hormonally regulated. And when sodium is reabsorbed, potassium is secreted. So again, that's hormonally regulated. In the collecting duct, we have water reabsorption that is not hormonally regulated. Water will just be reabsorbed through the process of osmosis and any aquaporins or transcellular routes that happen to be there. But it can be promoted with hormones. Those two hormones are aldosterone and antidiuretic hormone. Aldosterone acts to reabsorb sodium, water can follow sodium, and ADH acts to create more aquaporins so that more sodium can be reabsorbed. I hope that was clear for you, and with the other study materials you have a better understanding of reabsorption and secretion. Let me know if you have any questions. Thanks.